Electroporation is a biophysical process that utilizes short, high-voltage electric pulses to transiently disrupt the lipid bilayer of cell membranes, forming nanopores that allow molecules such as DNA, RNA, proteins, or drugs to enter the cell. In microfluidic devices, this process is integrated into precisely engineered microchannels where electrodes create uniform electric fields that can be carefully controlled. For example, a microfluidic chip may contain a narrow channel with integrated interdigitated electrodes that deliver a calibrated electric pulse, often in the microsecond to millisecond range, ensuring that each cell passing through experiences the same treatment. One may ask, why use microfluidics? The answer lies in the high precision and reproducibility offered by these systems. Much like using a finely tuned instrument to unlock the cell's membrane only temporarily, thereby minimizing damage while maximizing molecular uptake. Integrating electroporation with microfluidic devices offers significant advantages. The reduced sample and reagent volumes, combined with the ability to rapidly control reaction conditions such as temperature and ionic strength, result in highly efficient processes that require minimal waste. Imagine performing experiments in a highly miniaturized laboratory where every variable is precisely controlled. This leads to enhanced transfection efficiency and a dramatic improvement in the overall consistency of the results. In this context, the question, why not use conventional methods, is answered by the superior control and high throughput capabilities that microfluidic platforms provide, enabling researchers to fine-tune parameters that are critical for cell viability and treatment success. Microdroplets play a pivotal role in these systems by acting as individual isolated reaction chambers, encapsulating cells within microdroplets, each a tiny uniform volume of an aqueous solution suspended in an immiscible oil phase, ensures that every cell experiences identical electroporation conditions. In such setups, even small differences in pH, ionic strength, or temperature are minimized, leading to reproducible outcomes. For instance, water in oil emulsions generated in microfluidic chips can isolate single cells or small cell clusters, each undergoing the same electric pulse simultaneously. This isolation is similar to placing each student in a classroom in their own study booth, so that everyone receives the same attention and instruction, leading to uniform performance. In addition to microdroplets, microcapsules are also utilized in these applications. Unlike simple droplets, microcapsules possess a core shell structure wherein cells or therapeutic molecules are encapsulated within a protective shell made from polymers or lipids. This shell not only provides a barrier against external stresses, but can also be engineered to respond to stimuli such as electric fields. In practical terms, microcapsules can be designed to rupture at a precise electric field threshold, thereby releasing their contents in a controlled manner. One can liken this to a time-release capsule that opens only under specific conditions, ensuring that drugs or genetic material are delivered exactly where and when needed. The application of electric fields within microfluidic devices is achieved through the integration of specially designed electrodes on the chip. These electrodes, often made from transparent conductive materials like indium tin oxide, are arranged in configurations that produce uniform fields across the microchip. This uniformity is critical because it ensures that all cells, whether in microdroplets or microcapsules, are exposed to the same intensity of the electric field, minimizing the risk of localized overheating or overexposure. The design of these electrodes is akin to a carefully arranged lighting system in a photography studio where every subject is evenly illuminated, ensuring consistent and reliable results. Optimizing electroporation involves fine-tuning several parameters, including electric field strength, pulse duration, pulse frequency, and the composition of the surrounding buffer solution. Field strengths typically range from several hundred to a few thousand volts per centimeter, and the pulses are delivered for durations that are long enough to induce pore formation, but short enough to allow the cell membrane to recover quickly. Researchers often adjust these settings based on the cell type. For example, mammalian cells may require a pulse of 1000 V/cm for 0.5 milliseconds to achieve high transfection efficiency while maintaining viability. This process is similar to adjusting the settings on a precision instrument where every parameter must be balanced to achieve the desired outcome. The high degree of control provided by microfluidic electroporation significantly improves transfection efficiency. In conventional bulk methods, cells are exposed to uneven electric fields, leading to variable outcomes. In contrast, microfluidic systems allow for precise timing and dosing of electric pulses as cells flow through individual microdroplets or microcapsules, resulting in a higher percentage of cells successfully incorporating foreign molecules. This improvement can be compared to precision irrigation in agriculture, where targeted water application results in better growth compared to broad, indiscriminate watering. At the core of electroporation is the scientific principle of transmembrane voltage, dV. When the induced dV across the cell membrane exceeds a critical threshold, typically between 0.2 and 1 V, the lipid molecules reorganize to form hydrophilic pores. These pores are transient, closing once the electric field is removed. Detailed studies using high-speed imaging and fluorescent markers have shown that these pores form within microseconds and reseal shortly thereafter, a process that is essential for maintaining cell viability. This transient pore formation can be compared to temporarily opening a drawbridge, allowing passage and then promptly closing to protect the structure. Maintaining cell viability is paramount during electroporation, and microfluidic devices excel in this aspect by precisely controlling the intensity and duration of the electric pulses. Multiple short pulses rather than a single prolonged one are often used to allow the cell membrane to recover between pulses. Additionally, optimized buffer solutions and cooling mechanisms are implemented to minimize thermal damage, a strategy akin to giving a tired athlete short rests between bursts of activity, enabling sustained performance without fatigue. Buffer solutions and medium composition also play a crucial role in electroporation. These solutions are formulated to provide a stable ionic environment, maintain proper osmolarity, and ensure an even distribution of the electric field. A carefully chosen buffer can prevent excessive current flow, reducing the risk of Joule heating, and maintain pH levels that support cell stability. Much like preparing the perfect marinade for cooking, the right balance of ingredients in the buffer is essential for achieving optimal results during electroporation. When cells are encapsulated within microdroplets, single-cell electroporation becomes feasible. This technique allows researchers to isolate individual cells within their own microenvironment, ensuring that each cell receives a uniform and controlled electric pulse. This isolation not only minimizes variability, but also facilitates detailed studies on the cellular response to electroporation. Single-cell analysis can then be performed using techniques such as fluorescence microscopy, which provides immediate feedback on molecular uptake and cell viability, much like giving each student in a classroom their own personalized lesson plan for maximum effectiveness. Real-time monitoring is another critical component of successful electroporation in microfluidic devices. Techniques such as high-speed fluorescence microscopy, impedance spectroscopy, and real-time PCR are employed to observe the effects of electric pulses as they occur. For instance, fluorescent dyes that enter the cell only upon pore formation provide immediate visual confirmation that electroporation has occurred. This real-time feedback allows researchers to adjust parameters on the fly, ensuring that the process remains both efficient and gentle on the cells, much like a live sports broadcast that enables instant analysis and strategy adjustments. The applications of electroporation in microfluidic devices, especially when combined with microdroplets and microcapsules, are vast and varied. They range from gene transfection and targeted drug delivery to advanced cell therapies and high-throughput single-cell analysis. In cancer research, for example, electroporation can be used to introduce CRISPR/Cas9 components into tumor cells, leading to precise gene editing. Additionally, microcapsules loaded with therapeutic agents can be electroporated to trigger the controlled release of drugs at specific sites, minimizing side effects and enhancing treatment efficacy. This approach is revolutionizing personalized medicine by providing targeted, minimally invasive treatment options. Despite these advancements, challenges remain in implementing electroporation in microfluidic systems. Issues such as droplet coalescence, inconsistent electric field distribution, and channel clogging can compromise the efficiency and reproducibility of the process. Researchers are actively addressing these challenges through advanced microfabrication techniques, improved surfactant formulations to stabilize droplets, and computational models that optimize electrode configurations and predict electric field distributions. Overcoming these challenges is crucial for the development of reliable, clinically applicable devices. Looking toward the future, the field of electroporation in microfluidic devices is poised for significant innovation. Advances in automation, high-resolution imaging, and artificial intelligence-driven real-time data analysis are expected to enhance these systems further. Fully integrated lab-on-a-chip platforms that combine cell sorting, electroporation, and downstream molecular analysis are on the horizon, promising rapid, personalized diagnostics and therapies. One can envision a future where a compact device automatically adjusts electroporation parameters based on live cellular feedback, much like a smart device that continually adapts to its environment to optimize performance. This evolution has the potential to revolutionize healthcare, making advanced cellular therapies and precise genetic modifications accessible in a clinical setting. In summary, electroporation in microfluidic devices, especially when combined with the use of microdroplets and microcapsules, represents a powerful and versatile tool in modern biomedical research and clinical applications. Through precise control over electric field parameters, reaction environments, and real-time monitoring, these systems offer enhanced efficiency, improved cell viability, and the potential for high-throughput applications. As researchers continue to refine these techniques and overcome existing challenges, the integration of electroporation with microfluidic technology promises to open new frontiers in gene therapy, targeted drug delivery, and personalized medicine. Do you have any further questions, or would you like additional details on any specific aspect of this fascinating technology?